Hi everybody, this is the second PowerPoint for cell structure. The first PowerPoint we focused on the structures associated with the plasma membrane and how molecules and components can get in and out of the cell either through active or passive transport. The focus of this lecture will be on the structures that are present within the cell and the function associated with each structure. Now, the space that is present inside the cell minus the nucleus is referred to as the cytoplasm. So cytoplasm is the space and all of the organelles and the gel-like material that are present inside the cell. Now, if you take a look at this space, which is the cytoplasm, it is filled with a gel-like material, which is mostly water, and it also contains protein, salts, and sugar, and that gel-like material is referred to as cytosol. And within the cytosol, you can find components known as inclusions and organelles. Organelles are basically insoluble molecules that are present within the cell, such as glycogen, granules, pigments, lipids, droplets, vacuoles, and crystals. And when we talk about organelles, we're basically talking about the structures that carry a very specific function within the cell and can have a membrane, a plasma membrane around them, which we refer to them as membranous organelles, or if that membrane is absent, we refer to them as non-membranous organelles. So let's take a look at the different type of cytoplasmic organelles that are present. Now, if an organelle or a specific organelle within the cell carries a plasma membrane around its structure, we refer to it as membranous, and if that plasma membrane is absent, we refer to this as non-membranous. Presence of membrane allows for compartmentalizations of the structure within the cells, and again, any of these organelles will carry a specific function within your cells. So, if you recall from the definition of the cytoplasm is the space within the cells minus the nucleus. So, nucleus is a component of the cell that carries the genetic information either in the form of DNA or RNA, and it's also known as the control center. Majority of the cell in your body will carry one nucleus per cell. However, cells such as your cardiac muscle or your skeletal muscle will have more than one nucleus because of their length and their ability to basically provide the cell with enough control. Therefore, cells like skeletal muscles and cardiac will contain more than one nucleus. Now, when we're talking about nucleus, there are structures within the nucleus that are also need to be known. Since nucleus contains the genetic information, there is a membrane that protects the genetic information from the outer environment, and this membrane is referred to as nuclear envelope. It is a double membrane, but it does contain tiny little gaps referred to as nuclear pores that allows the genetic information such as RNA to exit the nucleus and enter the cytoplasm. You also, within the nucleus, if you think about this purple structure as the nucleus, within the nucleus you also have a secondary structure referred to as nucleolus. And the job of the nucleolus is to manufacture components of an organelle referred to as ribosome. So ribosome, as we'll talk about this, is responsible for protein synthesis. Lastly, we're talking about the genetic information that is present inside the nucleus in the form of DNA. If DNA is not being used during the process of mitosis, DNA can be found in combination with proteins, and we refer to this as chromatin. However, if a cell is undergoing cell division, DNA needs to be condensed and it forms structures known as chromatids. And if you put two sister chromatids together, then you create a structure referred to as chromosome, which is represented in typical text in a form of an X. So if we're talking about, again, these three components, so I have chromatin, which is uncondensed. Sorry, my handwriting is not the best. My computer has a mind of its own. So it has uncondensed genetic information. If we're talking about chromatin, the keyword is uncondensed genetic information, which is combined with proteins. Now, if I start to condense genetic information, it will form into chromatids. And if you combine two sister chromatids, what you get out of it is a chromosome. Again, sorry. So each of the drawings that I'm doing right here, this is one chromatid, this is another chromatid. The whole structure together is referred to as chromosome. And if this genetic information is uncondensed, we refer to it as chromatin. So what you actually see as genetic information will not be visible if a cell is not undergoing cell division. Here is a picture of a chromosome. So you see the X pattern represented here, and this is one chromatid, and this is the second chromatid. And the combination of both will create your chromosome. Now, discussing the structure we mentioned earlier in association with nucleolus is a structure called ribosome. Simply put, ribosomes are a form of RNA, which is referred to as ribosomal RNA or rRNA, and they are made up of two subunits, and these subunits are going to be basically combined together to form one full-size ribosome. Now, these ribosomes can be free-floating within the cytoplasm, and they're referred to as free ribosomes. And if those ribosomes are attached to a structure known as endoplasmic reticulum, we refer to those as membrane-bound ribosomes. But again, the goal of the ribosome, the function of the ribosome is protein synthesis, and in a lot of cases, they refer to them as protein factories. Endoplasmic reticulum is stacks of membrane that are connected to your nuclear envelope. So they originate from there. Now, if this endoplasmic reticulum contains ribosomes on their surface, they refer to as rough ER because ribosomes create little bumps on the surface of the endoplasmic reticulum, creating a roughened patterns, and therefore we refer to this as rough ER. The function associated with rough ER is it has an active role in protein synthesis, and it also enables your proteins to be folded to their secondary, tertiary, and quaternary structures if necessary. When the protein is folded and is ready, it gets delivered to the next structure inside a vesicle, and within that vesicle, you're basically going to have multiple proteins, and these proteins are going to be delivered to the next structure called the Golgi Apparatus, and I will talk about that in just a moment as well. Now, if we have endoplasmic reticulum, but there is no ribosomes attached to its surface, we refer to that as a smooth ER. So a smooth is lack of ribosome create a smooth surface. The function associated with the smooth ER include synthesizing phospholipid bilayer components such as your phosphate and fatty acid tails, as well as cholesterols and carbohydrates for your plasma membrane. It also has other functions depending on the specialized cells. It has the ability to break down your glycogen, which is a complex form of carbohydrates, to simple molecules of glucose. It also has the ability to detox your drugs, your carcinogens, and pesticides, if you're talking about a smooth ER present in cells of your liver and kidney. And in the case of your skeletal muscles and your cardiac muscles, these smooth ERs are specialized to carry a significant amount of calcium for muscle contraction. Here is a depiction of what the endoplasmic reticulum looks like. This part of the blue structure has the ribosome attached to it, so these would be considered to be rough endoplasmic reticulum. And this side you have no attachments, so no attachments of ribosome, so we refer to these as a smooth ER. Now, as I mentioned, when proteins are made, when lipids are also made, they get packed into the vesicles and sent to the structure known as Golgi Apparatus. So what Golgi does to these proteins and to these lipids are the job is to modify, concentrate, and package these components for the final delivery. A lot of times we refer to Golgi Apparatus as the FedEx of your cell, which basically receives your package, make sure it's packaged properly, make any modification that is necessary, and then eventually deliver it to its final destination. Now, the process is three-step process. First thing is you have the vesicles that are originating from ER are going to merge with your Golgi apparatus. The components of the vesicles come into the Golgi. Proteins and lipids are the main components. They can be modified, they can be tagged and sorted and eventually packaged. And your Golgi apparatus eventually send that package as a separate vesicle to any place that protein or lipid is required. Here's a picture of the Golgi apparatus and the process I'm describing. This is a vesicle coming from your endoplasmic reticulum. You see it being merged with your Golgi apparatus. It goes through the Golgi apparatus and eventually comes off as individual vesicles you see right here. So what is the destinations of these packages or these vesicles from your Golgi apparatus? So there's three separate pathways for these proteins and lipids. And they're represented right here, one right here, and one right here. The first option you see right here is when the proteins and lipids are merged with the plasma membrane through the process of exocytosis, and the components gets released into the outside. So basically you secrete the molecules or proteins via the process of exocytosis. The second option is you have components built into it, and those are going to basically be merged with the plasma membrane and be used as part of the plasma membrane. And the third option is that those proteins and components are going to be vesicles and proteins that are being used directly within the cell itself. So your options again, release them outside, embed them into the plasma membrane, or use them within the cytoplasm. Next structure we have is called mitochondria. As you can see, one of the most important components or characteristics of the mitochondria is the presence of the double membrane. So the inner folds are the first membrane, and then you have the outer fold or outer membrane. The job of the mitochondria is to produce ATP from your food or from your nutrients that you have consumed. And for that to happen, you do require oxygen, and you carry out a reaction referred to as cellular respiration. Fascinating thing about mitochondria is that it contains its own DNA, RNA, and a ribosome, which means that it can make its own protein depending on its need. So mitochondria is a very independent structure that is responsible for production of energy within the cell, but it also has the ability to make its own proteins. Lysosome is a structure that looks like tiny vesicles packed with digestive enzymes and an acidic environment produced with hydrochloric acid. And its job is to merge with other vesicles and break down any component that is unwanted by the cell. Now that could be to break down pathogens that could cause harm to your body. It can be large molecules that have come in through the process of endocytosis and will be broken down, or it can recycle any damaged organelles that needs to be removed. Any of the waste products produced as a result of this is eventually gets released through the process of exocytosis. Interestingly, lysosomes also have the power to destroy the whole cell through a process called autolysis. If there is a damage to the cell or the cell is infected, there is a possibility that the cell undergoes self-suicide or self-destruction by basically bursting the lysosome within the cell and causing the digestive enzymes and the hydrochloric acid to break down the cell from within, causing its death. Next, we have preoxisome. Preoxisome is another structure that carries enzyme within it, but the most important enzyme that is carried inside of it is referred to as catalase. Catalase is an extremely important enzyme because it takes something that is harmful to us, which is hydrogen peroxide, and it converts that to water and oxygen. Hydrogen peroxide is a result of or a byproduct of our metabolism. So hydrogen peroxide is, apologies again, my computer has a mind of its own. So hydrogen peroxide is H2O2. This is a 2, not a 3. And when it is interacting with catalase, it becomes H2O, which is basically water, and it's combined with O2, and oxygen. So preoxisome is extremely important because it takes something that can cause harm to our body and converts it to something that is required by our body. As a secondary function, by the way, it also has the ability to break down and synthesize fatty acids. Now this part is focusing on a structure called cytoskeleton. Cyto implies cell, a skeleton means basically the skeletal, right? Skeletal system, scaffolding of your body. So these are a skeletal within the cell. There are 3 different sizes: microfilaments, intermediate filaments, and microtubules. And their name basically describes their thickness. Microfilament is the thinnest, followed by intermediate, and microtubule is the thickest structure. And all of these are basically proteins that are combined together to form different structures. Now, where we can find microfilaments, as you can see in this picture, they basically spread out through the cell. This is fluorescent microscopy that helps you identify the structures within the cell. So in this case, they're highlighting the microfilaments, and you notice that microfilaments, again, it's spread pretty much throughout the cell. It has multiple functions, including a role in changing cell shape, the process of endocytosis and exocytosis, and movement of the cell throughout the organism. Intermediate filament, as I mentioned, is the second thickest, or second thinnest, depending on how you look at it. As you see that throughout the structure, it looks like robes, and the job that it carries, it provides some level of stability for your cell. It is relatively tough, and it helps maintain the shape of the cell, it stabilizes the location for the organelles, and the position of the cell within the organism, or within the tissue. Microtubules, each the thickest of them, as it says the word tubules, there is a cavity at the center, and it's made up of many individual subunits. The function associated with the microtubules include providing a strength for the cell and anchoring some of your organelles. It helps with shaping the cell shape. It's able to move the vesicles from one location to another. And during the process of mitosis, it acts as a spindle to allow for the separation of the genetic information between 2 cells. Lastly, within our structure, we have a non-membranous organelle referred to as centrioles. Centrioles are still made up of microtubules. They are present in pairs, and they are housed in a structure referred to as centromeres. The important function associated with this structure is to form spindles that, as I mentioned, plays a role in cell division in the process of mitosis, and it also can be used to form cilia and a structure called flagella that are essential for movement of the cell. Inside our body, we have both cilia and flagella, but flagella is more specifically associated with sperm cells, and cilia can be found in multiple structures, including your digestive tract and your respiratory tract. So that's it for this PowerPoint. We'll pick up the next lecture, mitosis, in the next presentation.